Okay. Thank you very much. Thanks a lot to the organizers for the organization of the conference. It's very enjoyable. I'm Luka Vucinic, and I'm a lecturer at GCU London, so satellite campus of Glasgow Caledonian University. And obviously, I'm neither from UK or Ireland, but I spent most of the last decade in Ireland, and this is part of my PhD that has been concluded several years ago. And obviously, Lawrence has been my supervisor. David, who just spoke, was also involved, as well as Catherine Coxon, who's not here today but sending greetings. A lot of karsts in Europe, I would say, especially in the region that I'm from originally, so Balkan region, from the Daneri karst. And when I decided to become hydrogeologist, I was obviously born and raised in Montenegro, where there's 2/3 of country is karstified. So being hydrogeologist from Montenegro means that karst, working on karst hydrogeology, is unavoidable. So it's not a passion or anything, it's a destiny. And then life took me to Ireland, where there is also a lot of karst, slightly less than in Montenegro, but not less exciting, I would say even more exciting. And in Venetian Italian, Montenegro means Black Mountain. So obviously it's not lowland karst that I encountered in Ireland, which is mostly lowland karst. And I plotted some of those sites that I've been mostly working on, so 9 karst springs in the west of Ireland, 4 in County Clare, 2 in Galway, and 3 in County Mayo. And there are some other sites that you will see in the next slides, where I also conducted some tracer tests that are hopefully interesting. And what I was primarily interested in was contamination from human wastewater sources, particularly septic tanks, for the reason that there are plenty of those in Ireland. About 1/3 of the population is relying on those. Here in England, I think about 300,000 to 500,000 dwellings are using these decentralized wastewater treatment systems. In Scotland, it's about 177. So percentage-wise, because population is smaller there, that's a higher percentage, but still significantly lower, higher than in England, but lower than in case of Ireland. And I've been looking at the sites, obviously karst ones, where with high vulnerability, where the karstified rocks are either exposed on the surface or very near the surface. And obviously in those kind of conditions, septic tanks usually do not perform well, but they can fail for other reasons as well, such as lack of maintenance and things like that. So what I was primarily interested was to tackle some of the challenges, at least partly, and I wrote them here in kind of two big, broad sentences, I would say. So I was interested whether we can understand better the origins of pollution, because a lot of vehicle pollution can come from the agriculture in rural settings. So we want kind of from the, I suppose, catchment management perspective, understand where the predominant pollution comes from. Try to quantify those different pollution impacts, which is obviously very important. And I was kind of interested in understanding better pollution transport, especially when it comes to microbial transport in the karst subsurface. And there are some graphical abstracts that I drew, okay-ish, I would say, from some of the papers that have been already published. So these are some of the papers that are out. And what I'm going to do today is going to run through some of these techniques that we used and share with you some insights found that are predominantly related to lowland karst. They might be slightly different in some other karst settings, but overall the key insights are, I would say they would remain, they would stand. So flow cytometry is a technique that has been used since around 50s and was used in biomedical research, and they were just analyzing blood cells. But then, especially in the last few decades, it's been more and more used in environmental research, and especially in water treatment. Why? Because it's very quick. And the technique that we used firstly was technique based on using those two dyes, where it's not like injecting huge quantities of dyes like in actual tracer tests. It's like very small concentration in the samples that we actually analyze with flow cytometers. And one of those dyes will enter in cells, in bacteria, whether they're damaged or not. But when you have damaged cell, which is compromised membrane, then obviously one of those dyes, such as propydium iodide, thiazole orange, which is parent compound, will enter in both. Propydium iodide would only enter in those with the compromised membrane and won't enter in intact cells. So that gives a flow cytometer that's based on lasers, and a lot of detectors it's kind of telling them to each detector how to detect and enumerate them. And yeah, it's very quick, easy to learn, easy to do. And firstly, what we did on 9 Karst springs in the west of Ireland, we tested what kind of insights we can get from measuring total cell counts. So all intact, live, or those damaged and dead, and what we can get insights from those ones that are intact, so without compromised membranes. And also there are some based on low nucleic acid and high nucleic acid content. We can kind of distinguish. They're broadly telling us how complex those cells are, so whether we can get some of those insights. And what we found is that flow cytometry can actually help us a lot to understand the pollution dynamics at Karst springs and in those catchments. But we also found that there are stable bacterial communities already present in Karst aquifers, those kind of simple environmental organisms that are kind of making our interpretation a bit trickier and more difficult. And we also obviously used some more conventional microbial analysis and evaluated as well turbidity, looked how those concentrations vary with different temperatures, etc. So again, this is just to kind of show to demonstrate what flow cytometry can do, but the details obviously are much more in detail in the paper that is already published. When I was very young, more than 20 years ago, when I started studies, hydrogeology studies at the University of Belgrade, but there's actually a program when you decide to study hydrogeology, you actually go directly to a program that makes you study hydrogeology from the bachelor level. And I was reading a paper where Croatian, former Yugoslavian scientists carried out tests with yeast, tracer tests with yeast, long time ago, maybe now 100 years ago. And that stuck with me. And once I started using flow cytometry, I was like, okay, we have differences in how solid dyes are being transported in the subsurface, Karst subsurface, and particles such as microbial organisms. So I remembered yeast, and I remember that yeast comes in various sizes, that it's ovoid, kind of round to ovoid shape, and those sizes are covering all the sizes of pathogenic microorganisms that we're interested in. So when we inject them, they will travel quite similar to those. And so we did that, and we did this on 2 sites in Ireland, and we actually found out what we kind of already expected, that they are traveling at different rates. So we tested how flow cytometry will perform in terms of picking up those yeast that we injected. We also injected dye tracers so we can compare those travel times. And we also used particle counter, portable particle counter that is similar technology, just doesn't use the same detectors. So you don't need to do any dyes, you don't need to stain those cells. But unlike flow cytometry, it will pick up debris and all the other particles present in the water sample. So both of those, very briefly, both of those performed very well. This is one of the This is from Lochgur, actually, from one of those pool of water, which I don't speak Gaelic. I don't know what it means. David told it's something that but I can't translate it either way, so I don't know what it is. But we had multiple spikes over there, and this was quite from the low flow conditions, so it was very different than what David found previously during high flow conditions. So that was quite interesting, and we also found that yeast was detected before as fluorescent dyes, and then consequently together at the same times. So that told us that both flow cytometry can be used to pick up those particle counters as well, and that yeast is very good as a tracer. It's a funny story, if I have time. When I was buying a lot of quantities of yeast, I was also doing microplastics analysis, and then they came to Lawrence, someone from Trinity, because I was buying bottles for brewing bottles to sample for microplastics, and also a lot of yeast. So someone emailed Lawrence from Trinity College asking what Luka is doing. Am I brewing some alcohol? And then Lawrence was very funny, replied, "This wouldn't be such a bad idea." So, but before microplastics, we also tried methodology that has been refined by Lawrence and Lawrence's former postdoctoral researcher, and in regards to fluorescent brightening compounds. So they're also known as optical brighters. They are being added to our to washing powder to make our clothes brighter, shinier, etc. They are also in toilet papers because they also want those to be nice and bright and shiny. And basically, they developed really good methodology to test them very quickly, very cheaply. You don't need a huge amount of training to do those. So it's great. Nowadays, you can measure fluorescent brightening compounds with probes, but if you don't have those, you can sample your sample in amber bottles. Keep them in the dark, in the cold, and you'll be able to analyze them and do that. So these are some of the results, obviously, based on the signal detection and how strong signal is. Based on those ratios, we can actually see how frequent we have those fluorescent brightening compounds, concentrations, signals in this case, strong, and then that can help us to understand whether we have impacts of pollution from human wastewater. In terms of microplastics, when we did this, there were no studies with regards to microplastics in groundwater. Soon after we started this, Pano et al. In the US, they published their now widely cited paper. So we took 5 liters for 5 liters. Now I know this is probably too much, and especially if you do what I did, filtering those samples, picking them up, and individually analyzing those using FTIR. So that took me quite a lot of time. And because of those reasons, and then obviously microplastics are coming, huge numbers are coming from during the washing clothes. There was a study that somehow estimated that 35% of all microplastics in the sea that ends up it's coming from the washing machines. I have no idea how they managed to calculate this, but you know how it comes with those kind of big calculations on such a large scale. They can be misleading, but if it's true, it's quite significant. And then obviously fluorescent brightening compounds are being predominantly released in the environment from the same processes. So I'm interested not really in numbers of microplastics, but I'm interested in those as a tracers of pollution and of human wastewater pollution. So these are some of the results from 5 springs. I only sampled 5 for 5 months because there were so many samples and this took huge amount of time. But what I found was that the majority were fibers, which was consistent with what Pano et al. Found. Both studies, both this one and Pano, found clear and blue fibers as the most dominant color type, which makes sense from the perspective of pollution, of human wastewater pollution. And these are the types of microplastics that have been predominantly found. And this is also, I couldn't really compare that much because Pano et al. Used different technique. and they burned all their samples, so they didn't get really, they didn't get that kind of information. But if you do some simple statistics, you, even with such a low N value, you kind of see that you have positive patterns. I'm not going to say they're kind of that are suggesting all positive linear relationship, because that would be statistically incorrect, but some of them are statistically significant, and others are not because of the low N number, but there are some positive patterns that might suggest that they would be also having significant positive linear relationships if I had more samples, but yeah, I don't have. Fecal sterols and stanols, those would be all fecal matter would contain sterols and stanols. They differ from in terms of humans because we are omnivores, and we have certain type of bacteria in our guts that are breaking them down in certain ways, which is different from birds, different from domestic animals. So we need on Karst Spring to sample huge quantities of samples, 20 liters. They need to filter them and do all sorts of all sorts of extractions and to prepare them for the analysis using GCMS. But this is what you need to remember, that you need to have total sterol content above 2 micrograms per liter benchmark to be able to interpret them in a meaningful way. And this is how that looks like. Here, I plotted the total concentrations, and obviously most of those were below, even when I was collecting such a huge quantities of samples, so that's laborious, unpractical. And then you can see here, when you do those kind of interpretations for those ones that you could, you kind of see what would be human concentration in that sample versus all the other sterols that can be from wildlife or domestic animals, etc. So that kind of can help you to quantify, if it's possible, whether you have predominantly pollution from human wastewater or from maybe agricultural sources. So these are some general takeaways. So flow cytometry can be very useful for not just rapid assessment of microbial water quality, but also can provide some additional insights in terms of fate and transport of fecal pollution in karst aquifers, and also can detect microbial tracers, injected artificial microbial tracers at Karst Springs. The majority of microplastics can be linked to human wastewater sources based on their properties and detection, simultaneous detection of fluorescent whitening compounds. The highest detection rates, unsurprisingly, are in those catchments with high densities of septic tank systems and high percentages, and where those kind of systems are very close, within 200 meters, or at least one karst feature that might indicate direct pathway to the aquifer, such as Fallowhall, Torlock in Ireland, etc. Fluorescent whitening compounds can be successfully used, and this method that we use is very inexpensive and reliable and easy to learn, and fecal sterol and stanols can be useful, but if you can justify, because it costs a lot, takes a lot of time, and you need to do a lot of work to get those results. So if you can justify that, it makes sense to do it. Thanks.